Hi everyone, welcome to the latest instalment of the Kings Park Festival Science webinars. Firstly, we want to acknowledge elders past, present and emerging of the Whadjuk Noongar people as the traditional custodians of the land on which this webinar is being hosted. Hi everyone, I'm Lily.
Hello, my name is Milana, and Lily and I are both PhD students here at Kings Park. We're doing our PhD projects, which are about cryopreservation, and this our presentation today is gonna talk you through process of cryopreservation and what are we doing in our projects.
Enjoy.
Due to extensive land clearing for agriculture, invasive weeds, diseases, air and soil pollution, and climate change, there has been decline in plant diversity worldwide. Thereby, many of the preservation methods have been developed. One of them is in situ or on-site conservation, which basically means that we keep the species inside of their native environment. Next one is ex situ or off-site conservation, which can be considered as supplement for in situ conservation, because species in this type of conservation are not kept in the native environment. And in ex situ conservation, such as botanical gardens, we have live collections of the plant species, and worldwide there are approximately around 2,000 botanical gardens. Next type of ex situ conservation are seed banks, where we keep the plant material storage at minus 20 degrees. We can see in the picture, this is the seed bank in Norway, and other two pictures are seed bank in Kings Park.
Another method of ex situ conservation is tissue culture. So for those species that can't be stored in seed banks because they don't produce seed, or they produce seeds that are sensitive to desiccation, they can be grown in tissue culture. And tissue culture is growing whole plants on agar media in the lab, and these jars are kept in sterile conditions. And one drawback to this method is that because the plants are continuously growing, we have to subculture them into new jars every month or so, otherwise they'll very quickly die. So this is a lot of work and requires specialized training and a lot of time. And these jars can also be susceptible to contamination. On the other hand, another method of ex situ conservation that overcomes these issues is cryopreservation. Cryopreservation is the storage of biological material at negative 196 degrees Celsius in liquid nitrogen. And this method is a very efficient method of long-term storage, because at these low temperatures, no change is able to occur to the plant material. It also means that there's very low chance of further contamination to the material once it's in the liquid nitrogen. And with this method, we can cryopreserve multiple types of plant material, including seeds, seed embryos and shoot tips. But we mostly use shoot tips in our work. These are some of the species that we have in cryo storage currently at Kings Park.
As previously mentioned, the climate change and air and soil pollutions and other things which we are witnessing in this time, we must emphasize the importance of the cryopreservation as a method for conservation of plant species. Intensifying research efforts into cryopreservation as a conservation tool is vital for increasing the ex situ conservation opportunities for threatened species.
Most sci-fi movies make cryopreservation look very easy. If you cryopreserve Han Solo, you can thaw him out again in a few years and he comes out looking the same as when you put him in. But unfortunately, it doesn't work this way in real life, even with plants. So you will have noticed this if your fridge gets too cold and you've put some lettuce in there and it comes out looking like this because it's accidentally been frozen. So some species can survive just being straight away exposed to liquid nitrogen temperatures, but these are usually plants from really cold environments where they naturally have to survive freezing every winter. Obviously in Australia these species are few and far between. So we have to do something else to the tissues to get them to survive freezing. And this process is generally involving a few different steps, from excision of the material to be used, desiccation, cryoprotection, exposure to liquid nitrogen, thawing and recovery. And we're going to walk you through the steps of this process and explain how each step helps our plant material to survive cryopreservation. Before we talk about each of these steps though, I want to talk about ice. Ice crystal formation is one of the main things that we want to always avoid when we're cryopreserving plants, or we'll end up with something like the soggy lettuce. So what happens when water cools down is the molecules of water begin to move together into an organized structure. This organized structure is ice. And these structures can grow into crystals, which can puncture cell membranes and physically damage our plant tissues. Water is also very unusual in that when it freezes, it becomes less dense, so it expands. So for example, if you put a glass bottle in the freezer to cool it down, and then you forget about it, you might come back a few hours later to find that the glass is smashed because the bottle isn't large enough to hold the expanded water. So in the same way, if the water inside a plant cell freezes just like this, it'll expand and burst open the cells, just like with your glass bottle. So almost everything that we do in a cryopreservation protocol is to prevent this from occurring and to increase the plant survival after cryopreservation.
So the first step in cryopreservation protocol is excision. This is the step where we get our propagules, which will be put out through the cryopreservation protocol. And here we have the video of how we are getting the shoe tips, the small shoe tips from the plant material.
So in aseptic conditions, we remove the leaves from the plant slowly, and we're trying to remove as much of that excess tissue as possible to get down to the meristem. And that's because the meristem is where all of the growth after cryopreservation will come from. So everything else is just extra, and all we need to preserve is that meristem. And the smaller we can get our shoot tip, the better, because the smaller it is, the faster it will freeze, and that gives less time for ice crystals to form and puncture our cells. So this is quite a time-consuming process, and it varies between different species. This is a green kangaroo paw. But
whenever, even though we use different species, we are aiming to get to shoe tips which are basically size of 1 millimeter or so. So as you can see, it's pretty time consuming and energy consuming.
Yeah.
But it gives better results. So basically, here are the pictures before and after. We got the small little shoe tip with the apical meristem right here in the middle, and from that meristem we should get a new plant after the cryopreservation. Next step in cryopreservation protocol is a step of desiccation. So in this step, basically what we're doing, we're trying to remove as much water as possible from the plant tissue to avoid ice formation, which can be very deleterious for the survival of plant. By doing this, we are minimizing damages to the plant material. So basically, our desiccation media has a high concentration of different types of sugars, and these sugars, when shoe tips are put onto desiccation media, these sugars are making an osmotic pressure, and in that way making the water to get out of the plant cells and making them, let's say, dried out. And here in the picture we can see Arabidopsis thaliana shoe tips, which are placed on the desiccation media.
After the desiccation period, we then expose the plant material to a combination of cryoprotectants. This very thick, sugary mixture draws even more water out of the tissues, in the same way as the desiccation media. But these chemicals can also move the other way into the tissues, which helps to support the cell structure and prevent the cells from collapsing from too much water loss. They also do one more thing to prevent ice formation, which is promoting the formation of a glass or vitrification. The way that they do this is that once they are inside the plant tissues and the material is cooling down, they actually physically get in the way of the water molecules coming together to form the organized ice crystal structure, and instead forms a disorganized glass. And we can see this in the two vials on the right. The vial on the left is filled with normal water, and you can see that it's cloudy due to all the ice crystals that have formed. But the vial on the right, which is filled with cryoprotectants, looks like glass, as it hasn't formed any ice crystals. The one problem, though, is that at room temperature, some of the most effective cryoprotectants are also quite toxic to plants. So it's a delicate balancing act to incubate the tissues just long enough to prevent ice crystals, but not so long that the cryoprotectants can cause too much damage. We can reduce the impact of this toxicity by doing this incubation step at low temperature, having our shoot tips as small as possible from the excision stage to make the cryoprotectants diffuse through the tissue more quickly, and by making sure that our material is properly desiccated before it reaches this step, so that less water has to be removed from the tissue. So you can see that all the steps leading up to freezing are really important and have to be optimized before we go on to freezing. But if everything is optimized up to this point, hopefully our tissues are well enough prepared to survive the next step.
So our next two steps in the cryopreservation protocol are freezing and thawing. As mentioned previously, freezing basically means that we are exposing propagules to the liquid nitrogen. And what is important is when propagules are at the temperature of liquid nitrogen, they are cryostored, because all the metabolic activity is basically stopped. There are species which have been cryopreserved for over 30 years. And next step is rewarming of our plant material. What we're trying within our experiments is to rewarm the propagules as fast as possible to avoid damages, as in to avoid the recrystallization of the water when it gets to the temperature of 0.
So once the plant tissues have thawed, we place them into a washing solution, which is important for removing the cryoprotectants so that they don't damage the plant tissues. Then we place them carefully onto a special media containing sugar, nutrients, and particular growth regulators that help the plants to recover from cryo as best as possible. So similar to when you're trying to germinate seeds, you want to give them everything that they need and the perfect environment to give them a good head start. As you can see below, some of our shoot tips will hopefully survive, and we can reintroduce them to tissue culture, and if it's for restoration, eventually into the glass house and the field. However, Milena and I usually only get to see our plants grow for about 4 weeks, at which point we record the percentage survival to see how effective our treatments were. Because as you can see from the pictures, a lot of the time not all of our shoot tips will survive cryopreservation.
Again, every light has its shadow, so cryopreservation also has its own limitations. In the picture, we can see on the left picture, we can see live cells with all the different cell compartments or organelles. And next to it, we can see this integrated cell, which has been put through a cryopreservation protocol, and we can see that organelles and cell compartments aren't visible as in the left picture. Cryopreservation induces stresses such as oxidative stress, osmotic stress, dehydration stress. Additionally, it does cause damages to the cell membranes. We have to mention the phytotoxic effect, which is connected to the cryopreservation cryoprotectants, and this effect can be seen if there is a too long incubation in the cryoprotective solutions, or the concentration of the cryoprotective solution is too high. We will emphasize here oxidative stress because both Lily and I are doing our research about the oxidative stress and effects of oxidative stress on plant cells. So we'll know that plants are aerobic organisms which need oxygen in order to live, and plants are organisms which also produce the oxygen. But with metabolic activity that is happening within cells, with oxygen there are produced also the reactive oxygen species such as peroxide, hydrogen peroxide, and others, which can be seen in the picture. Chloroplasts, mitochondria, and other cell organelles do produce reactive oxygen species continuously. So we shouldn't consider these species as necessarily bad for plant cells. because they can also have the role of signals within the plant cells. What is important is that plant maintains the equilibrium of these species, and plant cells do that either by enzymes, which are in the within the plant cells, or by non-enzymatic system or antioxidants. And these antioxidants, well, probably you've heard that your nutritionists suggested to you to have a daily intake of plants which are rich in antioxidants. Two most common antioxidants which are present in the plant tissues are glutathione and ascorbic acid, or most commonly called vitamin C. My project is about the model plant species, Arabidopsis thaliana, and here we have the picture of the wild type plant. We have decided to do the project on this specific species because it has a completely sequenced genome, which basically means that we specifically know the order of the bases of a DNA molecule within this organism. Next fact about this plant is that there is a large library of mutants of this plant, which are not naturally occurring in nature, but are made by human. So we can actually do more thorough research of different metabolic pathways. Another fact is that wild-type Arabidopsis thaliana has shown itself as a highly cryo-capable species, which comes very handy when you have to do the cryopreservation and when you have very good survival and regeneration rates. Many studies have shown the importance of the antioxidants in the DNA integrity and the cell cycle function. What I'm going to do in my project is I'm going to compare the wild type and mutant plants in order to get the insight in the metabolic pathways and the genetic stability of mutant plants. So as I mentioned previously, mutant plants are not naturally occurring in the nature. People use it and people make them because they want to have much better understanding and have a better insight into which genes are important for which activity and response for environment. And here in this slide we can see Arabidopsis thaliana wild type, which is also called Col0. Next to it is CAD, that's a mutant which lacks, well, actually it has about 70% less GSH or glutathione compared to wild type. And the third picture is a picture of VTC mutant, or a mutant which has 75 to 90% less ascorbic acid or vitamin C. What we can notice is that mutants are much smaller than wild type plants. Their leaves are smaller and they have sort of yellowish color, which basically itself shows that these plants are already struggling with, let's say, like a daily life, and that's why we want to use them in the cryopreservation to see which genes will be activated or deactivated when when they go through cryopreservation. So cryopreservation induces production of reactive oxygen species, and this hyperproduction of reactive oxygen species happens during the whole process of cryopreservation. So at every step of cryopreservation: excision, desiccation, cryoprotection, freezing, thawing, and recovery. Hyperproduction of ROS molecules can lead to the DNA damage. This is bad because when there is a DNA damage, a normal processes such as replication, transcription, and translation into protein, these processes are affected and cannot happen like in normal cells. Also, there are different types of DNA damage, which we can see in the picture. There are single strand breaks, damaged base, double strand break, and others. In order to make cryopreservational protocol more successful, we need to have appropriate cryopreservation protocol, which will then minimize the oxidative stress and thereby reduce DNA damage in plants. And let's touch the previously mentioned antioxidants with mutant plants, which lack the normal levels of antioxidants within their tissues. Addition of antioxidants can lead to the better response of plants to cryopreservation, which can then lead to higher survival and regeneration. Here we have the picture of a gene network. As we can see, it's not only pretty, but we can understand that it's very tangled up. So what I basically want to say is that there is no a single gene that responds to some specific change in environments and some specific stress. All of these genes have inner connection, and they can respond. Multiple genes can respond to specific stimulus from nature. Why hyperproduction of reactive oxygen species or oxidative stress can also affect the genetic stability of plants. Shifts in the redox homeostasis or stressed disbalance of a normal cell can lead to the DNA damages we previously mentioned, which can then affect the gene expression, especially within the mutant plants, which are part of this research. Thereby, optimization of cryoprocedure is important in order to reduce or avert cryogenic stress.
So before I go on to talking about my project on plant mitochondria, I'm going to talk about human mitochondria. So when you breathe in, you absorb oxygen in your lungs, and this oxygen is transported to your cells and then to your mitochondria. In the mitochondria, this oxygen reacts with high energy molecules that you've absorbed from your food to produce carbon dioxide, water, and adenosine triphosphate, or ATP. And this ATP is the energy currency of the cell. So your body requires it to do everything from walking to digesting down to the tiny reactions that occur within your cells. And this process is conserved amongst almost all living things, including plants. And considering the importance of ATP for doing everything, I'm willing to bet that that includes recovering from cryopreservation. One difference between animal and plant mitochondria obviously is that rather than eating food, plants produce these high energy molecules through photosynthesis in the chloroplasts. So this is a mitochondria. This one's round, but they come in all different shapes and sizes. And you can see up the top where the black arrows are that there's an inner and an outer membrane to the mitochondria. And the inner membrane forms folds called cristae, which greatly increase the surface area of the inner membrane, which is really important because this inner membrane is the site where the vast majority of ATP is produced in the mitochondria. However, this inner membrane is also the site of a large amount of reactive oxygen species production. And these reactive oxygen species are produced in greater amounts when plants are under stress. And unfortunately for plants, they're not able to get up and move around and move away from stressful environments like we can. So they've evolved some special adaptations in their mitochondria to help to deal with this stress a little better. And one of these is that plant mitochondria have a cyanide-resistant pathway, which basically bypasses certain points of this process of making ATP, which produce ROS. So it doesn't produce very much ATP, but it can have a role in reducing reactive oxygen species. And some studies have found that in agriculture, plants that are exposed to stress have a greater activation of this cyanide-resistant pathway, and that this results in reduced reactive oxygen species. So as well as making ATP, mitochondria are also involved in making other molecules for the rest of the cell to use, and they're also involved in triggering programmed cell death. And programmed cell death is a natural process, but it can occur in excess when exposed to too much stress, such as during cryo protocols. And this can, if programmed cell death occurs excessively during cryopreservation, this can reduce our post-cryo survival. So, as well as ATP being important to fuel recovery after cryopreservation, mitochondria could be important in reducing oxidative stress during cryo, and they might be involved in programmed cell death. But overall, we don't really know much about how plant mitochondria function during cryopreservation because not much research has been done on this area. One study did find that ATP content in Arabidopsis, thaliana and potato shoot tips decreased at each step of the cryopreservation process. Two studies on orchids and Arabidopsis also found that cryopreservation caused a decrease in abundance of proteins that are important in the ATP production process. Some people have also observed misshapen mitochondria after cryo and after exposure to cryoprotectants, which, as well as maybe pointing to damage to the mitochondria, it could also be a symptom of programmed cell death. But it's not all bad, so some studies have found that the cyanide-resistant pathway might be more active in protocols with higher survival, and this was correlated with reduced reactive oxygen species damaged in these plants. Overall, though, these results show that mitochondrial function is most likely affected in some way by cryopreservation. and we want to investigate this further. So how are we going to measure mitochondrial function during cryopreservation? So we can take advantage of our knowledge of this equation from earlier, and we can measure oxygen consumption as a measure of ATP production. So we have a machine at Kings Park called the Q2, which can measure oxygen consumption over time. So in each vial in the Q2, we can put a shoot tip, and we can measure how quickly this shoot tip consumes the oxygen in its little vial, and we can convert this oxygen consumption rate to a metabolic rate. And then we can compare metabolic rates of our shoot tips when they are exposed to different cryopreservation treatments to see how these treatments are affecting mitochondrial function. So these are some preliminary results from a green kangaroo paw. So the metabolic rate is on the y-axis, and along the bottom are different steps of the cryopreservation protocol. The first bar is shoot tips that have just been excised and not exposed to any treatments. Secondly, shoot tips that have been exposed to desiccation. Then shoot tips that have been exposed to cryoprotectants but not liquid nitrogen. And lastly, shoot tips that have been exposed to the whole cryopreservation process. So as you can see, as the amount of stress increases from left to right as we go along the cryo process, the metabolic rate drops each time. So there could be multiple reasons for this occurring. One reason could be that there's programmed cell death occurring at each step. It could be also a reduction in mitochondrial function at each step. But basically with this method, it gives us a nice broad overview, but we can't really see exactly what's happening to the mitochondria. So further down the line, we want to use some other methods to validate these results and use methods with greater sensitivity to see how different aspects of the mitochondria are affected at different steps of cryopreservation. So cryopreservation has been successfully used on many species, but not all. So we want to increase the amount of species that we can use cryopreservation on through gaining a greater understanding of plant responses to cryopreservation through our research.
And by using our research as a tool, we can optimize the cryo protocols for all the future plant species which are going to be put through the cryopreservation protocols.
Okay, so we have one question about, I'll just read out the question. Can you please explain what shoot tips are, and is there a particular age of the shoot tip that gives the best cryopreservation result? So shoot tips are basically, they differ between species where you find them on the species, on the plant. So with these green kangaroo paws, you would find them, I don't know if you can see that, but you would find them down at the base. Whereas if you had something like a grevillea, you would find them at the top of the plant where all of the new leaves are coming from. So basically a shoot tip is tissue that includes the apical meristem, and this meristem is made up of basically rapidly dividing undifferentiated cells. So they're a bit like stem cells, they can form whatever structure the plant requires. So it's this property of the shoot tip that makes it perfect for cryopreservation, because it can regenerate into a whole plant once it survives cryo.
Yeah, and regarding the particular age of the shoot tip, so in my project, I'm using Arabidopsis, which when I plant the seeds, I let the seedlings to grow for about 10 days. And after that, I'm doing the excision of the shoot tips, which are basically 10 days old.
But with other species that are growing in tissue culture, like the green kangaroo paw that I work with, the best time to isolate the shoot tips is 21 days after the last subculturing. And it normally occurs, well, the optimum time is normally around the 19 to 21 day mark for most species, but it does vary a little between species. There was also a question for me on why did you decide to work on the green kangaroo paw? One of the reasons is that it is a little like Arabidopsis actually, in that it grows really quickly, so it's easy to work with. And another thing that we found with it is that it gives a really good metabolic rate, which makes it easy to work with the Q2. And another thing is that, I just thought of something and I forgot. Oh, it has great cryopreservation survival. So if it doesn't survive cryo, it makes it really hard for us to see how effective our treatments are, because we don't have any data to work with. So if we have a species that survives really well, it'll give us better data for our research.
This question is for me. With the Arabidopsis mutants, do you expect them to survive cryopreservation? Well, as I mentioned previously, because of the low levels of the antioxidants, they will really struggle with the cryopreservation protocol. And the thing I'm going to do within my research is going to try to optimize the cryopreservation protocol. And by that, I mean I'm going to add antioxidants to the media on which plants mutants are growing. So I will see if those antioxidants will help mutant plants to better survive cryopreservation.
And one last question, I think, is about whether photosynthesis interferes with the Q2 results. So logically, you would think this because plants are obviously normally producing oxygen as well as consuming it. But normally with plants in tissue culture, because we're giving them all the sugar they need in their agar media, they don't really photosynthesize as much as normal plants. Plus, we're removing pretty much all of the green tissue from our shoot tips by the time they're on the Q2. And the Q2, we've actually tested the light levels at the top of the Q2, and it's very, very low light. So we don't think that it's possible that the shoot tips will be photosynthesizing as well as respiring. But I think we'll leave it at that for now. Thank you for listening again, and tune in next week for Russell Miller's talk on fire ecology in the Banksia woodlands and some of his work that he's doing here at Kings Park. Thank you.
Thank you.
Bye.